So we left off, this is the second video concerning proteins. We left off talking about this process of dehydration synthesis, which starts which bonds two amino acids together. Okay, so what happens on a bigger picture here? First of all, we know these polypeptide chains, as they grow, they're going to end up similar to how DNA has a 3' end and a 5' end. This is the polypeptide chains are going to have an N terminus and a C terminus. All right. So N terminus is the NH2 end. C terminus is the COOH end. The repeated sequence is NCC for the backbone of this, and you can see it's N for an amino acid, NCC, 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 and of course you have your C terminus, which I'm circling here, and your N terminus, which I'm circling over here. Now, look at the R groups here. This one actually has, they're not labeled as R, it has different amino acids here. You'll see here's an R group. Here's an R group. Another R group. And these are unique amino acid systems. Amino acids can only grow from the C terminus end. This is where new amino acids are added, and they can only grow in that direction. Right? So new amino acids will be added here. That's why they are numbered this way too. Notice there's numbers on all these 5, 10, 15. So the function depends on the structure of these things, right? They have a 3D structure. They are twisted, folded, coiled into a unique shape. This here again is hemoglobin. Hemoglobin. And this is collagen over here. Collagen. So this affects what they do. If you look at the hemoglobin, you can tell that this is a very kind of circular, globular structure. The collagen over here is formed in these kind of straight lines. It's 3 separate proteins kind of bonded together to give strength. So this is a long structural protein. So typically, when we talk about proteins, we are going to talk about different levels of structure. And I like to think of it as zooming in. And there are 4 of these levels, which we'll talk about starting with the next slide. 4 levels of protein structure. So there is primary structure. Primary structure, I like to think of this as you're zoomed in as close as you can to see the actual amino acids, right? These 3-letter designators are indicating which amino acids go where. Of course, what is that called? 3N. Should be calling that the N terminus, right? N terminus and the C terminus over here. C terminus. Okay, so your primary structure is simply the order of amino acids in the chain, right? The amino acid sequence, of course, is determined by the coding in your DNA. Slight change in the sequence can affect the protein structure and affect its function. For instance, if you know this amino acid right here is a, let's say, I don't know if it is or not, but let's say it's a hydrophobic amino acid. And you have a mutation that gets rid of this amino acid and replaces it with a hydrophilic amino acid. That can have a huge impact on how things end up here. So it's important to look at primary structure when we want to look at what happens when you change one of these amino acids. If you change the properties of this amino acid, it's likely to change how this protein functions, right? Ultimately. So that's primary structure. You're zoomed in very close and you're looking at the order of amino acids. Okay, secondary structure. Now we've zoomed out just a little bit, and we're going to look at the local, what we call the local folding. In other words, this is kind of the structures that are made at a local level. Folding along these short sections of a polypeptide. These are often formed by hydrogen bonding, as you'll see here. These are weak bonds. Hydrogen, of course, we know hydrogen bonds are weak, but I like to think of them as fairly strong bonds in a way as well. This starts to form this 3D structure, and there's 2 3D structures we talk about when we talk about secondary structure. This is what I want you to know. Secondary structure involves the alpha helix, which is one of these structures you're going to see on proteins. On almost every protein, you'll find some alpha helices. You'll also find these things called beta pleated sheets. Beta pleated sheets, which are these long, flat, folded, or pleated sheets of amino acids right here. Right? And you'll notice that these dots here, these represent hydrogen bonding. And typically these beta pleated sheets, they're going to form like this. You'll have a chain of amino acids that kind of comes back around like this. Right? And it has directionality to it. but it'll form this sheet. Of course, for your coils, you're going to have amino acids kind of coming around like this, and that still has that directionality to it, right? You would have a C terminus over here, and terminus over here. Here, C over here. But this is the secondary structure, right? This is zoomed out just a slight bit. Now, if we zoom out even further, and we want to look and make sure these are very important to understand, or at least acknowledge that they exist at this level, we're talking about protein structure. Zoom out just a little bit more, and let's look at the tertiary structure. This is the actual bonding, kind of how this whole thing folds together, and how it starts to look on a larger scale. There's interactions between distant amino acids, right? Things that aren't close by. If you'll notice, look at this interaction right here. You see this positive negative ionic bond being formed up here. Those amino acids probably aren't very close on this chain, right? But you can kind of start to see these interactions between the whole structure, not just this local folding that you see in here with this helix. Start to see how the whole thing folds together. So hydrophobic interactions are very important. Hydrophobic, if you remember, is water-fearing, right? Water for hydrophobic. Obviously, you know phobia, you're scared of it. Water-fearing, that's how I like to think of it. Water-fearing interactions. Cytoplasm is mostly water. So nonpolar amino acids, which do not dissolve well in water, remember this one in class, like dissolves like, meaning that nonpolar stuff is not like polar stuff, right? So you're going to find a lot of these not hydrophobic interactions, these nonpolar R groups. You're going to find that they like to face each other, and they don't like to face towards the outside of the molecule where all the water is. So they're going to cluster away from the water. Hydrogen bonds and ionic bonding, you'll find hydrogen bonds, in this structure as well, and ionic bonds, as we saw up here. Stop one. Disulfide bridges are a very special type of bond you're going to find between sulfurs. Usually, this is on cysteine amino acids. You don't need to know specifically about that. Just know that sulfurs can bond to kind of hold these amino acids together, or these polypeptides together. And I think that's it for tertiary structure. Let's move on to quaternary structure. So quaternary structure is more than one polypeptide chain bonded together. So in other words, this over here, again, we've seen this one. This is kind of the classic example here. This is hemoglobin. It has 4 different proteins right here, 4 different subunits. They're different colors over here, varying shades of like orange and pink. These are all separate proteins, but they happen to be built separately, and they bond together as part of their process. This is hemoglobin. This is a quaternary structure. This means that there's more than one polypeptide chain, and it's only when they're gathered together does it become functional. So these things cannot function on their own. They must be pulled together as hemoglobin. Again, here we go with some collagen. Collagen has these 3 separate polypeptides that are kind of wound together for strength. This for strength. You'll find these in your skin and tendons. Hemoglobin you're going to find in your blood. So let's go ahead and summarize everything we know about proteins. The monomer for proteins is called amino acids, right? There are 20 unique amino acids. We need to know also that they have a amino bonded to central carbon, bonded to a carboxyl group, carboxyl. So there's 20 unique ones. The polymer for proteins is called a polypeptide. It has a primary, secondary, tertiary, and quaternary structure. Know what those are. Again, I think of them kind of as zoomed in to zoomed all the way out. Peptide bonds. This is what we call the bond between amino acids. There's a covalent bond between an amino and a carboxyl group. You'll find these in every protein. You're going to find this peptide bonding. It's what gives proteins this chain structure, right? It is these peptide bonds that can create this backbone. The function of proteins, they do pretty much everything. They're the jack of all trades. They can speed up chemical reactions. They can contribute to the structure of a living thing. They can transport things. They can communicate. They can operate in defense. They can help in movement, as in your muscle tissue. They operate in growth and repair. So this is pretty much all you need to know about proteins for now. We're going to talk about this more in depth later in the year. But for now, I think this is going to suffice. So thank you for listening.